Assalamu alaikum wa rahmatullah. I will discuss the membranous organelles and the cell membrane in this section. This is a molecular structure of the cell membrane. This picture showing the molecular structure of the cell membrane. The cell membrane is formed of three characteristic molecules. The first molecule is called phospholipid bilayer. The phospholipid bilayer is present in two layers. The one layer is formed of globular phosphate head, which is hydrophilic, and fatty acid tail, which is hydrophobic. This layer is present as two layers, which is called phospholipid bilayer. The second molecule of the cell membrane is the protein molecule, and the protein molecule is present in two types. The first type is called the transmembrane protein or the integral protein, which passes through the phospholipid bilayer and it functions as a channel. The second type of the protein is called the peripheral protein, which is present in the periphery of the cell membrane and doesn't pass through the phospholipid bilayer. It functions as a receptor. The third molecule is called the carbohydrate molecule, and it is present in the form of chains, which is bound to either lipid molecules, which is called glycolipid, or protein molecules, which is called glycoprotein. This is a mistake. This is a glycoprotein. This is also the picture of molecular structure of the cell membrane. When this picture comes in the exam, I will write molecular structure of the cell membrane. This is the phospholipid bilayer, the integral or transmembrane protein, and it is the peripheral protein. This picture showing the cell membrane by EM. The cell membrane doesn't appear by LM because it is very thin. It appears by EM as trilaminar appearance, which is three lines, two lines with electron dense, which is a deep gray, and the middle line is electron lucent, which is light gray. This picture showing two cell membranes with a lower magnification. On higher magnification of these two cell membranes and these two cells, they appear as two cell membranes with trilaminar appearance, and the space between the two cell membranes is called intercellular space. This is the cytoplasm of this cell, and this is the cytoplasm of the another cell. So when this picture comes in the exam, I will write two cell membranes of two adjacent cells. Two cell membranes of two adjacent cells. This is the intercellular space. This is the intracellular spaces. This is our folds of the cell membranes, which appears as finger-like projections. This folds of the cell membrane is called microvilli. The function of the microvilli is to increase the surface area of the cell, which is present in the small and large intestine, to increase the surface area for absorption. This fuzzy-like material on the cell membrane is called glycocalyx or the cell coat, which is formed of the glycolipid and the glycoprotein molecules, which is the carbohydrate portion of the cell membrane. The glycocalyx or the cell coat is concerned with cell adhesion, cell immunity, and cell recognition. The first membranous organelle is called mitochondria, and the word membranous organelle means that this organelle is surrounded by a membrane, The mitochondria is a membranous organelle. It is surrounded by two membranes. The outer membrane is smooth and doesn't form folds, and the inner membrane forms folds, which is called keraste. The inner folds of the mitochondria of the inner membrane contains globular structures, which is concerned with ATP synthesis. So the function of the mitochondria, it is the powerhouse of the cell, which is concerned with energy production and ATP synthesis. So the inner fold is this inner folds is called keraste, and the fluid part of the mitochondria is called matrix. This picture showing multiple mitochondria with multiple keraste, meaning that this cell is a highly active cell. This picture showing mitochondria with a small number of cristae, meaning that this cell is a less active cell. This organelle is called RER, which will be discussed later. The second organelle is called rough endoplasmic reticulum. The second organelle is called rough endoplasmic reticulum, which is also a membranous organelle. It is formed of parallel channels. This parallel channels is called cisternae. The outer surface of these parallel channels is studded with electron-dense particles, which is called attached ribosomes. This organelle with its abbreviation RER can be written in the exam. This organelle is concerned with protein synthesis through the attached ribosomes present on its outer surface. The protein formed by the attached ribosomes get internalized into the cisternae of the RER, which is transferred then to the organelle called Golgi apparatus to be secreted as secretory vesicles. So the protein formed in the RER is immature protein, and it will be modified in the Golgi apparatus so it can be secreted in the form of secretory vesicles in the Golgi apparatus. So I will see in the rough endoplasmic reticulum parallel channels with outer surface attached ribosomes. This picture showing rough endoplasmic reticulum or RER. This picture showing RER and a mitochondria with multiple cristae. This is a cell called plasma cell. The plasma cell have a characteristic shaped nucleus. This is the nucleus of the cell, and this is the cytoplasm of the cell. This cell has a characteristic nucleus which is called a clock face appearance of the nucleus, and this is the nucleolus. The cytoplasm of this cell contains multiple mitochondria with RER, the rough endoplasmic reticulum, meaning that this cell is concerned with protein synthesis and secretion. The third organelle is called smooth endoplasmic reticulum with its abbreviation SER. This smooth endoplasmic reticulum is formed of branching and anastomosing channels or cisternae. Branching and anastomosing channels of the SER, when cut on transversally cut, they appear as circles of variable size. These circles doesn't contain attached ribosomes in its outer surface, so this organelle is called smooth endoplasmic reticulum. So the smooth endoplasmic reticulum will appear as multiple circles of variable size, and they doesn't contain attached ribosomes on its outer surface. This is the mitochondria organelle. This picture contains RER with its parallel channels and SER with its variable shaped circles and no attached ribosomes on the surface. This diagram showing the branching and anastomosing tubules of the SER and parallel channels of the RER, which is studded with attached ribosomes. The Golgi apparatus, which is also a membranous organelle. The Golgi apparatus is concerned with protein secretion. The Golgi apparatus it's formed of convex surface, which is called cis face. Cis face because it receives the RER from the protein from the cisternae of the RER. Receive protein from cisternae of the RER, so it is called cis face. It receives the proteins in the form of vesicles, which is called transfer vesicles. Then the protein present in the transfer vesicles will enter in the sacules of the Golgi apparatus. The channels of the Golgi apparatus is called sacules, which is formed of narrow middle part and dilated outer end and dilated outer part. Narrow middle part and dilated peripheral part. This channels is called sacules. Then the protein will be modified in the sacules of the Golgi apparatus, and it will exit at the trans phase, a trans phase which is the mature phase of the Golgi apparatus. It will be exit from the trans phase of the Golgi apparatus. This concave surface is called trans phase in the form of secretory vesicles, and it will be secreted in the cell. So the cis phase side contains transfer vesicles, and the transphase side contains secretory vesicles. This is a diagram. The EM picture of the Golgi apparatus, I will see this is the cis phase, which is the convex phase, and it contains transfer vesicles. This is the channels of the Golgi apparatus, which is called sacules. This is the concave surface of the Golgi apparatus, which is called transphase, and this vesicles is called secretory vesicles. This is also a Golgi apparatus, and this is the convex surface, which is the cis phase. This is the sacules of the Golgi, and this is the transphase. This picture also showing Golgi apparatus with a convex surface, which is the cis phase, and this is the transfer vesicles, and the concave surface, which is the transphase, and this is the secretory vesicles. This is two cell membranes of two adjacent cells. This is a cell, and this is also another cell, so it is a two cell membranes. This also the Golgi apparatus. The Golgi apparatus doesn't appear with the routine H and E staining, so I will prepare a special stain to appear the Golgi apparatus on the cell. On staining the cell with H and E, the Golgi apparatus doesn't appear surrounding the nucleus, and it is called a negative Golgi image. So I will prepare a special stain for the Golgi apparatus to appear in the cell. This is an organ in the male genital system called the epididymis. The epididymis is formed of tubules. These tubules, when cut transversally, they appear as rounded circles. This circles is lined with epithelial tissue. When taking this part of the circle and a higher magnified it in this picture, this appear as that this part is lined with epithelial tissue. This is the nucleus of the cells, and this brown mass present above the nucleus, which is called supranuclear, is the Golgi apparatus stained with silver stain. So the Golgi apparatus appear in the epididymis above the nucleus, which is called supranuclear, and it appear as a brown mass. So the special stain of the Golgi apparatus is called silver stain. So this picture when come in the exam, I will write Golgi apparatus in the epididymis organ present supranuclear, and between the two brackets I will write the silver stain, which is the special stain of the Golgi apparatus. It is very, very, very important to write any special stain in the exam. This special stain have half of the mark. Finally, the final membranous organelle is called the lysosome. The lysosome is the digestive apparatus of the cell. The lysosome come from the Golgi apparatus. The first lysosome, which come from the Golgi apparatus and doesn't involve in any digestive event, appears as circle with homogeneous electron dense and surrounded by a single membrane. Homogeneous electron dense in shape, a homogeneous electron dense circle surrounded by a single membrane. This is called the primary lysosome. Also, this picture contains the primary lysosome, homogeneous electron dense circle surrounded by a single membrane. When the primary lysosome eat anything in the cell, it will transform it into secondary lysosome. There are three types of secondary lysosome. The first type, according to the vesicles that the lysosome eat, the vesicle which is called phagosome, which contains bacteria or viruses, or in generally any solid particle. The phagosome when fused with the primary lysosome, it will be transferred into heterolysosome. So the heterolysosome is a type of the secondary lysosome, and it appears in the cell as circle with electron dense areas and electron lucent areas, which is called heterogeneous. So the heterolysosome appear as heterogeneous circle with areas of electron dense and areas of electron lucent. So when this picture come in the exam, I will write heterolysosomes, not secondary lysosome. The type of the secondary lysosome should be mentioned in the practical exam. When the primary lysosome fuses with apinocytic vesicles, apinocytic vesicles is a vesicles contains fluids, and it will be transformed into multivesicular body, which is the body contain multiple vesicles of fluids, which is called pinocytic vesicles. When the lysosome fuses with old organelle like mitochondria, it is called autolysosome. So the autolysosome is the third type of the secondary lysosome, which fuses with the old or worn out organelles in the cell. This is also a secondary lysosome, which is called autolysosome, fuses with old mitochondria in the cell.